Okay. Yes. Please remember that all the review also I will put up the PPT. So if I didn't share it, please remind me to do that. Okay. So in today's class, we are going to discuss the structural dynamics of DNA, topology, and electrophoresis. So first thing, we will try to understand what is denaturation and what is degradation. So if you had a DNA molecule A, T, G, C like this, one strand, the complementary strand would have been T, A, C, G, right? There will be two here hydrogen bonds, three hydrogen bonds like that. So we should always mention 5 prime to 3 prime. So the opposite would be 5 prime to 3 prime. Anywhere you're drawing diagrams, you should always have these the polarities and the end groups mentioned clearly. So if this DNA molecule becomes separated, A, T, G, C, and the bottom one, C, G, A, T. Whenever you're reading, you should always read from 5 prime to 3 prime. That is what I'm trying to do here. So this is one double-stranded DNA molecule. And now it has separated into two single-stranded DNA molecules, right? These two are complementary in nature, but then they have been separated. That is referred to as denaturation. Okay. If you put these two nearby, they will again form the hydrogen bonds and reform the double-stranded DNA. That would have been described as renaturation. I hope you understood denaturation and renaturation. Denaturation is a reversible process where if you increase the temperature, the hydrogen bonds could be easily broken, because of which these strands would be separated. But if you reduce the temperature, the hydrogen bonds can be formed again and renaturation would occur. So there is another word, degradation, when you are using in terms of nucleic acids. Degradation would mean that you have a breakage in the phosphate backbone. Whenever you have a breakage there, you call it as degradation. It is an irreversible process. You need enzymes and energy to make it up, to repair such kinds of breaks. If a DNA molecule was like this, which became into like this, that is degradation. If the same DNA molecule became something two single strands, that is denaturation. I hope you will be able to use them appropriately. So here is something re-annealing. Renaturation, you can call it as re-annealing as well. And there is also something called hybridization. If you have two different DNA molecules, say I will put TGACT, okay? I am base pairing with another DNA molecule. So TAGTGT, GA. Are there, can this hydrogen bonding happen between A and T? Yes. T and A? Yes. G and G? No. These two can base pair, these two can base pair, and these two can base pair. The information on these two DNA molecules is not exactly same. I'll try to draw a little better, hopefully, so you will understand a difference between what is and what is not genetic information. Just give me a minute. Okay, so I'm trying to redraw the structure that I was trying to explain you. So I'm going to have two DNA molecules, A, T, G, A, C, T. What would be the complementary of them? T, A, C, T, G, and A. assume that's one DNA molecule. I'm also going to draw another DNA molecule, which will have almost the same sequence: A, T, C, A, C, T, T, A, G, T, G, A. So if you see these two DNA molecules, the information between these two DNA molecules is very similar, but not identical. Similar means you share a lot of properties. Identical means it's exactly same. Okay? So the difference is somewhere here, right? This is the difference. So you can say they are highly similar because they can have a lot of hydrogen bonds. If you have a situation wherein you have T A G T G A and second and another you have denatured it using temperature, and you have put denatured, these two would have separated, denature would have happened in this strand, in this DNA molecule. The second one would also have denatured. And if you allowed them to mix up, and then what would happen is something like this. What may happen, it's not that it has to happen. Say if you had 10 molecules of DNA here X and 10 molecules of double stranded DNA Y, you denatured and mixed them, then hybrids like this will form. When do we use the word hybrid? That's the question. When you have two complementary DNA molecules which are not absolutely, which is formed because of renaturation or reannealing, and the information is little bit or something is different, then you call it as hybridization. Okay, so here this is something that is different. One strand from here has G here, the complementary of which would have been also has G. So this is a hybrid molecule. Hybrid for what? Both are DNA molecules. They are hybrid for the information. Okay. So always keep in mind that all of molecular biology is studying about information part of it. Yes, absolutely true that the information is in the physical or chemical form, but what our focus is on the information part of it, because the if the a cell has to, a protein has to form, a cell has to form, or an organ, anything should happen, everything is coming from the genetic information. So when we study molecular biology, we should see that we are studying the nature or the information technology of life, or you're just studying bioinformation. Okay, when you see that, you there is a new different sense of it, because it's fully about logic, and information is of course logic too, logic as well. So I hope I have addressed the terms: what is denaturation, what is degradation, renaturation is or reannealing is same, and what is hybridization. So here we'll go by what is given in the textbook, a nice a simple experiment like thing. Say this is a wild type gene. You can call this part as wild type. And the other one is somewhere, the difference between these two DNA molecules is that in one place there is one gene that is present in the wild type, whereas in the mutant that A is missing. Okay. Are these two similar, wild type and the mutant? Yes, they are very similar because the rest of the information is same in both. Are these two identical? No, they are not identical because the wild type has gene A, whereas the mutant is missing that. So there are two different molecules. The information, genetic information is different. So when you heat them, the double-stranded DNA will become single strands like these, and when you put them back into normal temperature and cool it slowly, they will try to find, you may have wild type strands coming together and they may form. This is not a hybrid, not a hybrid. You may also have mutant strands coming together and base pairing, and that is not a mutant either, or not a hybrid, sorry. But if you have one from here and the other from here, there are two strands coming from two different, containing two different molecules, information, sorry, then that is a hybrid. So we have two hybrids here, this one and this molecule, because there is a difference in their genetic information. So that is what will happen, and the extra pair or the gene would usually remain like that. Okay, I hope you understood these things, dynamics. It will be useful in understanding, especially when we come to about when come to discuss about mutation, repair, and so on. These kinds of information or the terms are important. The the denaturation curve, which is a nice experiment actually, if one is trying to do. The first thing you should all know is DNA or nucleic acids absorb at 680 nanometers. Okay, why do they absorb? Usually that is an assignment question. Why do nucleic acids absorb maximum at 260 nanometers? And for your information, this is for nucleic acids. Um, and proteins would have absorbed at 280 nanometers. Okay, remember that. 260 for nucleic acids, 280 for proteins. That's where the absorption is maximum. It is usually because of the ring structures and the groups present in the the nitrogenous bases that absorb at 260 nanometers. So here are some more things that we need to understand. Hyperchromacity, there is some melting temperature, TM, and so on. So we are trying to understand this graph. On y-axis, we have absorbance, the amount of absorbance at 260 nanometers. And on the x-axis, we have temperature in degrees centigrade. So at 30 and 40 degrees, it will remain as double-stranded. But I mean, it depends on many things, including if there is a DNA molecule which has more of GC, and if there is the same similar length in size in base pairs, but it's rich in AT. Which one do you think will require more force to separate these two? The one with more GC or the one with more AT? Anybody? I hope you understood the question. If not, I will try to re-say. So there is a DNA molecule. It has 100 base pairs. And there is another DNA molecule, which is also 100 base pairs. The only difference between these two is assume that this is made up of completely GC only, and the other DNA molecule is made up of only A and T. So which one do you think will require more energy for denaturation? Yes, anybody? So what is denaturation?
The GC one will require more energy.
Yeah, why is that?
It's a
Um,
triple bond, right?
Yes, Aditya, you're saying?
The GC is a triple bond as compared to the AT, which is a double bond.
Okay. Just to correct, okay. What is a triple bond and what is a double bond? Or I will put it like this: two hydrogen bonds and double bond. What is the difference between these two? I mean, just it's a semantic or a chemistry wise of a way of determining. If you have carbon carbon like this, that is a double bond, right? One atom making two bonds with another atom. This would have been a triple bond. One atom making three bonds with another atom. In case of hydrogen bonding, we should not use the words double bond or triple bond. Just for a correction. I hope you understood that. In hydrogen bonding or anything, you have H, one hydrogen being in link with or sharing something with another atom. And you have another H somewhere else, which will have another hydrogen bond. One atom, one atom. Here, double bond, if you are saying, two bonds between two atoms. Triple bond is this. So we call these as two hydrogen bonds or three hydrogen bonds. We do not say, we should not use the word, I mean, double or triple, just for information. Okay, but the answer is correct. If you say 100 base pairs, you have assumed that these are all Gs. You have 100 Gs. And they would have base paired with Cs using three hydrogen bonds each. So the number of hydrogen bonds between these two strands would have been 300 hydrogen bonds. Whereas is if you had in the second case, which is AT, you have 100 A's and 100 T's, which would have been 200 hydrogen bonds. So it is easier to break these 200 compared to that of 300 hydrogen bonds. So you need less energy here, whereas in the other case, you need more energy. So there are many things. Just for what we are trying to here understand about one particular term that's called as hyperchromacity. Hyper is something like more, and chromacity is here, it is about absorbance. Okay. Somehow, I hope you can also guess why the single strand DNA can absorbs more than double stranded DNA. Okay. Double stranded is somehow something like this. They are hydrogen bonds. Some of the groups and other things that are present in the ring structures of the nitrogenous bases are engaged in hydrogen bonds, and they are inside in between these two. Whereas if they became single stranded, they would have been something like this separately, right? In that case, because now there are a lot more free exposed groups that can absorb more of the light at that wavelength. So single strands always absorb more than the double strands. You have to remember this. Say I took what I was talking here. I said 100 base pairs. In total, I have two strands, and just putting for understanding sake. But both these strands are in double-stranded nature, so their absorbance is less. Whereas if it is still 100 base pairs, but they constitute totally of two strands, but they aren't single stranded nature, then in that case, the absorbance is usually high. So the double-stranded DNA will remain as such until a temperature. But once the transition temperature is reached, it will start denaturing slowly. The hydrogen bonds that are present between these will start breaking up. And at one point, all the hydrogen bonds would have been broken, and all of the DNA would have become single-stranded DNA. Okay? And from then on, there is no change, so they will remain in this at this level. I mean, the absorbance will remain constant. The midpoint of this transition from double-stranded to single-stranded is referred to as the melting point, or TM simply. Okay, it is the midpoint of transition between double strand to single strand. What is it measured as? It is measured as the temperature. So if I have to define melting point, midpoint or melting point from here, it is like the temperature at which 50% of the DNA is in single stranded and 50% would have been in double stranded nature. That's how you can also look at it. I hope you understood a few things I hope you will remember those. Um, I want you to also have a way of calculating. There is a formula. There are on the internet you will find multiple formulas, more than one at least. So in all these cases, this is theoretically, if you knew the composition of a DNA molecule, then you can calculate the theoretical melting temperature. There will be deviation from it a little bit due to many other factors, including the buffer and other things. But if you are doing PCR and other thing, PCR, some techniques like that, you might want to know a theoretical value. For that, this formula is useful. So in this slide, I hope you remember what is hyperchromacity. The increase in absorption at 260 nanometers by single strand DNA. That's called as hyperchromacity. Midpoint or the melting point or TM is the temperature at which DNA a double-stranded DNA has denatured. 50% of double-stranded DNA has denatured. That's how you can also see. Or you can say the sample has 50% in single strand or denatured and say 50% in double strand nature, something like that. This is there are I was actually already describing the GC content, the effect of GC content. Now I would want to describe something about the salt concentration. I hope you will, I will ask a few questions. I hope you will be able to answer me. These are two strands, complementary strands, okay? 5' to 3', and in the anti-parallel, this one. So what are the forces that keeps these two strands together? You should tell me. We already discussed this. So quickly, if you tell me, it will be easier. The base pairing hydrogen bonds that are present between these two strands are the ones that are keeping these two complementary strands together. What are the opposing forces? You have to look at it this way. As I said, the backbone is made up of phosphates, right? The backbone is called phosphate backbone. It has, and there is negative charge on it, right? And if these two strands come close together, the negative charge on this phosphate and the negative charge on the opposite phosphate here that is the repulsive force, right? That is the one that is trying to keep the two strands separate because of the repulsion, because of they both have negative charges. Then, so now what do you think will happen? If I neutralized the negative charge here, what do you expect? Will it denature fast, or will it denature slowly? What will happen to its melting temperature? I will repeat the question if you didn't understand it. So I have described the attractive forces between them are the hydrogen bonds. The repulsive forces are the negativity in the between the phosphate atoms on the phosphate backbones of the two strands. So if I reduce the negativity of these phosphate backbones, what do you expect? Do you expect an increase in melting temperature or decrease in melting temperature? Anybody? So I will give some arbitrary, or just for understanding, I will just give some terms. So I hope you'll understand it then, hopefully. Let's see. Mm, yes. So I will say attractive forces are 10 units. I'm just giving it like that. Okay. The repulsive forces are 5 units. Okay. And then you would need 10 units of, you would actually require 5 units more of temperature to separate these two strands, isn't it? Because the other 5 is coming from the repulsive. From the repulsive, you have 5 units, and you are providing additional 5 units. The units of attraction are only 10 units. So if you match it, these two strands are likely to separate, right? Now, if I reduced or removed this repulsion, how much should I increase the temperature or decrease the temperature to separate these two strands? We have to provide, attractive forces are 10 units. Repulse, you are providing 5 units. There is no repulsion. So you have to provide more temperature to separate these two strands, right? So that is what salt does. When you add something like sodium chloride, the sodium ions can interact with these phosphate groups non-covalently. They interact with the phosphate groups, and because of this interaction, the negativity, that's electronegativity of this phosphate, is reduced. So what happens then? You have to provide more temperature or heat to separate these two strands. That means with increase in salt concentration, you will get the melting point would increase for that DNA. I hope you understood this point. And the last one, I already told you, if you have more number of GC contents, then you have more hydrogen bonds. That means you would require more temperature to separate those two strands. The second one, in terms of salt concentration, that is shown in green here, where if you add salt, the electronegative forces that are there on the phosphate backbones are reduced, because of which you would have to provide more energy to break the hydrogen bonds, because the repulsive forces are eliminated because of the salt concentration. So you can see it in those slides, and I hope you understood the graph. All of science is about one handling the data of that respective science. Here we are going to have a lot of tables and other things. You should know what it represents and what is presented and how to interpret that data. That's important for anybody aiming for research. Okay. So what you see here with GC content, you will see that with higher GC content, the melting temperature increases. And if you increase, say, if you increase the salt concentration in which it is present, the DNA is present, then also you will have more of the melting temperature increases. DNA topology, topology is about how it is organized in a higher level than coils. Okay, so there is something called as CCC, covalently closed circular DNA. Majority of the plasmids and bacterial chromosomes, they are circular in nature, something organized like this. In the next class or so, I will try to play a video, or I will send you a link of it. I would want you to watch those videos and we can discuss later. We are not going into a lot of detail in this case, but we will understand some fundamentals as much required. So these are the number of bases. It is showing five turns, and so on like this. how many turns are there. Okay. Each time one strand crosses over another strand like this, that is called linking number. Okay. Sorry, that is called twist. Okay, I made a mistake. That's called a twist. Twist, and when you have one double-stranded DNA molecule like this, go over another itself like this, that is something like right, which is represented as WR. So keep that in mind, and I would want you to turn on your video now, and I want you to hold on the rope. Please do that. Turn on your camera, please. Yes, if you can see me, I hope you can. Am I visible?
Yes, sir.
Okay. If you have a rope or something like this, can you please hold it on? Yeah. And assume this is a double-stranded DNA. And I
Okay.
want you to rotate on one side. Keep one stable, and the other one, I want you to rotate.
What?
Go. Yes, keep rotating it on one side. And I hope you can see the structure that is forming in my hand. Right? Do you see that? A structure has formed. Can you see my? Yes, keep rotating. And so basically, if this was a double-stranded DNA, assume it is already coiled. You turned it once. That means you increase the topology by one linking number, one twist. And you keep doing it, and you will form some other structures forming like this. Okay? That is called as supercoiling. Everything that is made of something physical like this, if you keep on rotating, it will form something. You are all aware of what happens with the case of your earphones and so on, right? This is called, each time one goes on top of other, that is equivalent to right. There are two different types. I used to have a different one, and I'm unable to have it. So there are two different types. You can see it here, interwound and toroidal. I'm just increasing, and some structure is forming, and that structure is interwound type. There is another structure that can actually form. You need to have something like that. If I put my finger or something and I keep twisting, the wire will keep on going on top of it. So let's just see what's happening on top of it. If I keep rotating on one side, this will form a spring-like structure around this pen here. And that is nothing, that is similar to that of toroidal. Okay, I hope you understood what is twist. Every time one strand goes on top of the other, that is a twist. Every time a DNA molecule or something goes on top of another like this, that is right. Okay, and when you increase the twist, the structure is getting much more coiled and something like this. That is supercoiling. Okay, all DNA is present as supercoiling DNA within cells. Okay, you can turn off the video now. I hope you understood. I would want you to experiment a little bit and have a look at what all structures can form. So with supercoiling, there are two, right? One is interwound, something like this, and the other one is toroidal, which is something like a spring. If you look at old landline telephone cords, this is a typical view that you will usually see. 1 second. Okay, and there is, if you had 10, if you had one helical loop, what is the twist of it? What is one loop? That is one DNA going around, one strand going over another strand. One loop is equal to one twist. Okay? So if there is a DNA molecule and it had 10 loops, what would its twist be? Twist would have been 10. If I took a DNA molecule which has 10 loops and rotated on the one side like we did in the little while ago, that means we are increasing the twist of it. Okay? And that is when the Wright or supercoiling actually happens. If you increase, so let me get it again. 10 loops will form 10 twists, will have 10 twists. If you increase the twist or decrease the twist, there is a stress on the backbone, because of which it tries to form supercoiling structures. I will take up topoisomerases in the next class. I would want to discuss a little bit about electrophoresis. Okay, at topoisomerases we will discuss or deal with it later. I hope you know electrophoresis. I'll quickly go through it so that you will remember it. The practical aspects we will learn when we are doing the lab. Here we are going to just deal with the theoretical aspects of it. So typically you have a gel and it is in a tray, and you will see all those things later. So DNA is positively charged or negatively charged? Can anybody answer?
Negatively charged.
Okay, that's right. DNA is acidic or basic?
Acidic. It's an deoxyribonucleic acid. So
That's right, acid. Because many people will, including me, we put a lot of emphasis on the nitrogenous bases, and some people will tend to say it as basic, but it's actually acidic. Why is it acidic? It's because of the phosphate groups that are present in the backbone. That's highly electronegative, and that's why it is negatively charged. All nucleic acids are negatively charged, and they are acidic in nature. So if I want to separate them, and electrophoresis is based on, it is about mobility of charged particles in electric field. That is electrophoresis basically. So you should have, this is negatively charged, and I'm going to put the DNA molecules here, and assume this is 100 base pairs and the other one is 1000 base pairs, and I want the DNA molecules to travel this way. And what charges should I put in the electrodes here and here? How should the electric field be? The DNA is already negatively charged. It will only move towards the positively charged. Therefore, the electrode here should be positively charged and the electrode here should be negatively charged. Then when it is connected to the electrical field, DNA molecules will move towards the positively charged electrode. But one is 10 times larger than the other, say sample A and sample B. Sample B has the molecular weight of the DNA, or in terms of length of the DNA, is huge. So certainly the length, the mobility of 100 base pair, the smaller one, will be much faster. Rate of mobility will be faster compared to that of the larger one. So size of the DNA. Size of DNA is one thing. So the smaller ones usually travel faster. The larger ones will travel slower. The second way of thinking also is there. where I have given you two different samples. Both of them are, I'll call it as C and D. Both of them are 100 base pairs, 1000 base pairs in length. How will they travel? They should travel in equal rates, something like this. But now I add a difference. As we just saw in the topology, a DNA molecule can exist in multiple ways. I will repeat some parts of it in the next class because I think I should have discussed about relaxed DNA, and I hope you remember about what is supercoiled DNA. It is highly theoretical. For that purpose, I think you should have watched the animation before we have the class. But during the review in the next class, we will deal with that. So in the meantime, we'll have an introduction to this. So assume this is 1000 base pairs, and this one is also 1000 base pairs, 1000 and 1000. This base composition is same, length is same. What is different? The topology is the difference. The topology also has an influence on the rate of mobility during electrophoresis. Size is certainly there. What we have already established, that's true. The second part is topology. In topology, the more supercoiled, the faster it will travel. We put this in agarose gel. The agarose gel has tiny pores, okay? You will see it that are not visible to us. They will have, based on the concentration you take, they'll have tiny pores. The DNA molecule should travel through them, right? And if it has to travel, if it is much more compact, like in this case, in the supercoiled nature, then it can travel faster. So supercoiled DNA moves faster than less supercoiled one. And the linear one, then comes the linear one, then comes the relaxed one. Okay. So for some reason, the linear one travels faster than the relaxed DNA. Here is a nice experiment that is taken where you treat the sample with topoisomerases, which I said I will discuss later in the next class. I would want you to first have a look at the animations. Okay. I hope you understood electrophoresis. We will continue it in the next class with ethidium bromide. These are all important aspects that we will discuss in the lab as well. So please read the textbook. I cannot emphasize how important the textbook is, especially because I'm following the exact same textbook. Please try to read it, and then I hope you will benefit to the maximum from that. I'll give you the molecular weight of this one, 300 or 330 Daltons, say. AMP is, what is the molecular weight of a single strand DNA, which is composed of 300 adenylates? I'm not saying it's AMP or ATP and so on. So you can note this question and try to find it out. So if I gave you 100 ATP molecules, and then I told you to polymerize it into a single strand with all consisting of A's like this, what would be its molecular weight? I think the molecular weight of ATP is somewhere around 357 or so. I can't recollect exactly. It should be right. No. Sorry, this is wrong. Yeah, I got it wrong. It is 507 ATP. ATP molecular weight is 507, and AMP should be somewhere around 330. Here is something you should remember. Molecular weight of ATP is 507 daltons and AMP is 347. Can you solve this question? And then we will stop quickly. I think I passed the time. Anybody? So the first ATP would be like this, right? And then next ATP would come. I hope you remember I said beta and gamma phosphates will be released out, right? So the next will be something like AMP, 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 and so on. Just a theoretical molecular weight can be done. So if you had 100 adenylates that have polymerized into one single strand DNA, the first one would have been ATP. So you will have molecular weight of 1 ATP, 507 into 1, and the remaining are 99, that is 347 into 99. That should give you the total molecular weight of the single stranded DNA. In this question, the point is that you should know that beta and gamma phosphates of any of the nucleotides during polymerization are released out. They don't become the part of the nucleic acid. Only the alpha phosphate will become the part of the nucleic acid. Okay, I'll stop here. Thank you. Any questions?